Okay, can everybody see me? See the screen at least? Everybody happy? Thumbs up if you can see the screen. Yep, okay. Right, so this is going to be a little bit self-indulgent, not to say how clever I was all those years ago to discover brown fat uncoupling protein, but to give you an insight into some of the thought processes that go along with this sort of train of thought, and see if these are going to be of any use to you in thinking how one plans a project. One doesn't actually plan a project. The project appears out of nowhere and you grasp it. So my work on brown fat was in the 70s and 80s, basically, and it still continues to be active. And one can do my favorite trick, which is going on to PubMed. And I did a search a week or two ago for brown adipose tissue or brown fat, and I had 16,000 hits, 16,000 articles dealing with brown fat. To be more specific, going to uncoupling protein, abbreviated UCP, nearly 8,000 hits. So it's still a very active subject. And what I thought I would do is to go through the logistical steps by which we arrived at the uncoupling protein and how it was actually regulated. And for those of you who are not familiar with brown adipose tissue, here is a slide of the normal white adipose tissue adjacent to brown adipose tissue. White adipose tissue has a single large fat droplet, very few mitochondria squeezed around the side. Brown adipose tissue is brown because it's full of mitochondria. The fat droplets are small, so-called multilocular, which increases their surface area. And as I say, they're packed with mitochondria. And the most dramatic thing about brown adipose tissue is its thermogenic capacity. When optimally stimulated, it can produce heat at a rate about 60 times the amount of an equivalent mass of liver, for example. And brown adipose tissue for many years was a curiosity for rodents, but also for newborn humans, and then disappeared. And then some 10 years ago, people started finding brown adipose tissue in limited amounts in the adult human. And so the whole field was rejuvenated and became fairly hot, as it remained, literally, and no pun intended, as it continues today. So let's start with my favorite subject, thermodynamics. And this is a model of a brown fat cell. What I want to emphasize is that the rate of heat production by any mechanism is dependent on the rate of conversion from substrates to products, how fast that occurs, and is totally independent of what's happening inside the black box. So it's independent of mechanism. Now, the problem one is faced with in brown fat, as you realize from the problem-solving problems that you've been solving the last couple of days, is that under most conditions, you are very careful to use the minimum amount of substrate. So rate of substrate oxidation is subject to respiratory control. And bearing in mind this slight leak here, it means that the rate of respiration is the minimum required to produce the amount of ATP that the cell needs. So it is ATP turnover which is controlling the rate of respiration. And if we start thinking about this, this creates a problem. And the problem is that you could have as many mitochondria as you like. They could be as active if you like. You could have as much triglyceride hydrolysis, lipolysis as you like. But if the mitochondria are still constrained by respiratory control, you would have a very limited heat production, and so the tissue would not work. So somehow one has to override respiratory control. And there are three ways of doing this. One can hydrolyze ATP rapidly by some extra mitochondrial mechanism. This is called shivering. So in shivering, the muscle fibers undergo, as you will appreciate, rapid oscillations, which is there to hydrolyze ATP to phosphate, allowing the mitochondria to work more rapidly, allowing them to produce more heat. So that is shivering thermogenesis. A second possibility is to modify the electron transport chain, to have an electron transport chain which either does not pump protons or pumps fewer protons. And one example is to bypass complexes 3 and 4, for example, with an alternative oxidase, feeding from ubiquinol down to oxygen, but not pumping protons. And this is an option which is taken up by various plants, for example, the arum lily. This lily, I forget what this bit of a lily is called now, but it gets hot. You can see on a thermal camera, it gets up to 32 degrees above the ambient 17 degrees by rapid mitochondrial respiration, not fully coupled to proton translocation, but with an alternative oxidase. So this would be another possibility. You could have mitochondria in brown fat, which have an alternative oxidase. The third possibility is to introduce an additional proton re-entry pathway. And this is the option which is adopted by brown adipose tissue. And I want to go through a series of experiments at how one arrived at this strange thing called an uncoupling protein. This is a brown fat mitochondria. It's actually a real an electron tomography reconstruction of a real brown fat mitochondrion, which I have sitting in my office at home, courtesy of Terry Frey. So this shows the outer membrane, the inner membrane, and the extremely extensive invaginated cristae. So an enormous surface area. And as we know, of course, all the exciting things are happening across this inner mitochondrial membrane. So brown adipose tissue look extremely active just looking at them, because they obviously have a lot of electron transport components. So if we go back, gosh, 70, doing my maths, if we go back 50 years, half a century, to the earliest studies on mitochondria from brown adipose tissue, and the earliest attempts to prepare brown fat mitochondria and to treat them as being similar to liver mitochondria, classic respiratory control, they all failed. The mitochondria were uncoupled. They show no respiratory control. And this was a question, are they damaged, or is it that these mitochondria are modified so they never show respiratory control? And the single most important finding was by Hans Raphael, who discovered people were thinking that fatty acids has something to do with it. Fatty acids can uncouple virtually any mitochondria if you give enough of them. He removed the fatty acids and you still can get respiratory control. And then he started thinking, maybe substrate level phosphorylation is involved. And in those days, we didn't know very much about permeability across the inner membrane. And so he said, let me add GDP or GTP to look for substrate level phosphorylation. He added the GTP, and miraculously, his mitochondria started showing respiratory control. Now, more or less at the same time, another group in Stockholm, a Lindeberg group, were again looking at fatty acid oxidation in brown fat mitochondria, and found that if you, instead of adding albumin, if you activated and oxidized away the fatty acids with CoA to form acyl-CoA, carnitine to get it into the mitochondria, and you also need ATP for the fatty acid activation. Once the fatty acids were oxidized away, the mitochondria showed respiratory control. So there appeared to be just fatty acid removal being essential here. It turned out that the ATP, which they were adding in order to get the fatty acids activated and oxidized, was having the same role as Raphael's recoupling nucleotide. So by the early 1970s, fatty acid removal and a curious nucleotide addition was necessary in order to show respiratory control. The problem was that in the early 70s, or late 60s, early 70s, nobody was talking about Peter Mitchell. Everybody was steeped in what I call the fog of the chemical coupling hypothesis. And one of the great advantages that you guys have had that I didn't have when I did my undergraduate degree in Cambridge was I wasn't exposed to the chemical coupling hypothesis, where a hypothetical high energy, the word I hate, high energy intermediate, which we abbreviate as squiggle, that squiggle was the intermediate for the production of ATP from ADP and phosphate, and that uncoupling meant the hydrolysis of squiggle. So it was totally impossible to get any mechanism out of this hypothesis. Their hypothesis was wrong, for example. So it remained a pure phenomenon. Now you might say, well, Mitchell had been around for ages, then why doesn't one simply read Peter Mitchell's ideas? And the problem is, this is the form that it was presented in the 1960s. Extremely difficult to understand. Peter was brilliant, but he made no concessions to normal mortals with normal intelligence in terms of his hypotheses. And so they were generally largely ignored for several years. And in fact, one of the classic mitochondriologists, Ephraim Racker, F. Racker, 1963, said, if you're not totally confused, you don't understand the problem, basically. So that was the state of the mitochondriology when we started looking at brown adipose tissue mitochondria to try and understand them. Now, there are two important things, important stages to make an important discovery in science. One is to be extremely lucky, and the second is to realize you've been extremely lucky. In other words, to say not my cells don't work or my mitochondria don't work, it's that this is very strange, my mitochondria are trying to tell me something. And this happened to me very fortuitously when I started working in Stockholm. And we were measuring the respiration of brown fat mitochondria oxidizing palmitoyl carnitine. It's a very good, palmitoyl carnitine is a very good substrate. And we prepare mitochondria in sucrose, the standard way of preparing them. And what we found, putting them into a normal KCl-based medium, the respiration slowly started increasing. This is the rate of respiration here. So this would be, these days, this would be a seahorse trace with steadily increasing respiration. They didn't respire in a sucrose medium. If one did the inverse, if you actually prepared them in KCl and put them into a sucrose medium, they went the other way. So the ability of the mitochondria to oxidize palmitoyl carnitine seemed to depend on the incubation medium. And what became apparent was that the mitochondria were permeable to chloride. Now, mitochondria are normally not permeable, or very slightly permeable to chloride, which is why you can use a KCl-based medium, which gives osmotic support. But this is repeating the previous experiment. This is the rate of respiration of sucrose mitochondria into KCl, into sodium chloride, which responds more rapidly, and then looking at the matrix volume. And the way you look at the matrix volume, you measure the tritiated water permeable 14C sucrose impermeable space. And the difference between the two, the water gets right into the mitochondria, the sucrose just gets across the outer membrane, gives you a measure of the matrix volume. And what we found was very rapidly in KCl, the mitochondria expanded. They swelled. They swelled much more rapidly in sodium chloride. And the conclusion was that we have two strange things about these mitochondria. They show no respiratory control unless you add a nucleotide at GDP, and they're permeable to chloride. So what do you do when you have two strange unknowns? You put them together and you ask the question, is there a link between the strange recoupling and the strange swelling? And a very simple way of measuring ion permeability is to look at the passive swelling of the mitochondria in isotonic KCl, for example. If you add valinomycin to make the potassium permeable, you're then limited by the chloride permeability. Now, somewhat counterintuitively, as mitochondria swell, they scatter less light. And the reason is that as they swell, the difference in refractive index between the medium and the matrix decreases, and that decreases the light scattering. So we'd imagine that bigger mitochondria scattered light more, but they actually scatter light less. And swelling requires charge balance. Cations and anions have to balance, and cations and anions must both be able to enter. So we did the experiment using two potassium ionophores, valinomycin, which is a potassium uniport ionophore, and nigericin, which exchanges potassium for protons. And then we looked at the swelling, and we found, first of all, that they swelled at the same rate in the presence of valinomycin or nigericin. And this tells you for the start, although we didn't realize it at the time, that the mitochondria are proton permeable. But when we add a nucleotide, GDP, we blocked swelling both with valinomycin and with nigericin. And so the conclusion is that GDP, among other things, is inhibiting chloride swelling, this curious chloride swelling. We could do experiments with potassium phosphate. And again, this is the iron shows with potassium phosphate. And then we could add albumin to remove the fatty acids. And what we found didn't actually fit with what we thought, because albumin blocked the swelling, which was reintroduced by FCCP, but we knew that albumin did not induce respiratory control. So things aren't fitting. If they had fitted, we would have come to this conclusion several years before. And the problem is that passive swelling is all very well, but it involves very low iron gradients, 10, 20 millivolts or so, low iron gradients, whereas the mitochondrion is working up close to 200 millivolts. And so it became apparent that what one had to do was to measure the proton motive force. It was this stage where I started my fascination with the electron circuit analog of the electron transport chain, and finally got round to reading Peter Mitchell, which I should have done 10 years earlier. And this is the figure I showed you in the beginning of the first talk, my coffee-stained copy of Mitchell's book, which I actually stole from a colleague. I cut off his name at the top out of embarrassment. But I realized one had to start looking now at the proton circuit in detail in the brown adipose tissue mitochondria. And to look at this mysterious leak. So to quantify it, we need to measure respiration, and we need to measure proton motive force. And Mitchell had a wonderful technique, which he published in the end of the 60s, using potassium and pH electrodes, and came to the conclusion that mitochondria in state 4 could maintain a little bit over 200 millivolts of proton motive force. It was extremely difficult to reproduce these experiments. They were very well done, but they were very time consuming. So we modified it to an isotopic experiment using rubidium, which behaves like potassium, for the membrane potential in the presence of valinomycin to make the rubidium permeable, and then the distribution of a weak base and a weak acid to give you the pH gradient across the membrane. And when we tried this out with rat liver mitochondria, we got a very nice trace. We had about 220 millivolts in state 4. We added ADP. We saw this slight dip in proton motive force, which recovered as the ADP was used up and converted to ATP. And there were the individual components of membrane potential and pH gradient. And when we now moved on to brown fat mitochondria and did the same idea of measuring membrane potential, respiration, and working out conductance by Ohm's law, we found that because we have two variables, fatty acids and purine nucleotides. The fatty acids can be present or not, and the nucleotides can be present or not. So you have 4 possible combinations of activators and inhibitors to look at. And I'll just focus on the proton conductance. We found that in the presence of fatty acids, the proton conductance was vast. We couldn't maintain any proton motive force across the inner mitochondrial membrane. It was almost as though the inner membrane was totally open to protons. And if we removed the fatty acid with albumin, the conductance was still too high. We could only generate about 80 millivolts of proton motive force. And we ultimately had to both remove the fatty acids and have the nucleotide present to reduce the proton conductance close to the basal conductance common to all mitochondria, and to get a proton motive force high enough for respiratory control. So that was a basic idea of adopting the chemiosmotic theory, the proton circuitry, and taking the brown fat mitochondria into the chemiosmotic era and actually measuring directly the proton conductance of this unknown mechanism in these different conditions. And you can do really elegant and physiologically irrelevant experiments. You can take brown fat mitochondria and you remove the fatty acids, and if you remove the fatty acids, they only maintain about 80 millivolts of proton motive force. And then you can start titrating in the inhibitory nucleotide, GDP. And as you do it, the voltage across the membrane increases, the current, the respiration, decreases, and applying Ohm's law, you see this beautiful hyperbolic relationship of the conductance of the unknown conductance pathway as a function of GDP concentration. And this shows you actually something rather important, which doesn't turn up with a lot of research which is done on brown fat. And this is that you only get information out of an oxygen electrode experiment if proton information on proton permeability, if proton permeability is rate restricting. And it turns out that you have to inhibit the uncoupling pathway by about 70 or 80% before you show any respiratory control. So anything with a conductance above about this level does not show up on the oxygen electrode, but you have to use both the voltmeter and the ammeter in order to get the detailed conductance out. So, and this has proved extremely useful, this idea of measuring conductance in this way. So what does the GDP do? The GDP binds to a high affinity site on the outer face of the inner membrane. It's non-covalent binding. It's relatively high affinity, a few micromolar. These are things called Scatchard plots, where you plot bound against bound over 3. The slope gives you the affinity, and the x-axis gives you the capacity. So this tells you that there are about 0.7 nanomoles of GDP binding site per milligram of mitochondrial protein. And this binding is unique to the brown adipose tissue. So we're beginning to home in on what is actually causing this. And the way we did this was to use a photoaffinity label. So a photoaffinity label means this one is azido ATP, which has an azido group on the end. When you flash it with powerful ultraviolet light, the nitrogen N2 falls off and you're left with the extremely reactive nitrine radical, and that binds to the first thing that it sees. And so first of all, one has to prove that the azido ADP works like GDP to inhibit respiration. It does. You have to show that it competes for GDP to binding, that it does. So the azido ATP is going at least partly to the same place that the GDP was going. So things are now getting exciting. We're getting close to a mechanism here. And in those days, one did tube gels. One didn't do high resolution flat gels. And by making the azido ATP radioactive with P32, we found a tightly bound doublet with molecular weights 32 kilodalton and 30 kilodalton. The 30 kilodalton was abolished in the presence of carboxytetractylate. Carboxytetractylate binds and inhibits the adenylucleotide translocator. And so we concluded that that site was the adenylucleotide translocator. If you have carboxytetractylate and GDP, you get no binding at all. So by the process of subtraction, this is the GDP binding site, and that was the physiological uncoupling protein. And nice work, for example, from Martin Klingenberg's laboratory a few years later showed that the 32 kilodalton protein was necessary and sufficient to act as an uncoupling protein. So it was a single 32 kilodalton peptide, which was acting as the uncoupling protein. And we have no crystal structures yet, but it has very close homology to the adenylucleotide translocator, and various groups, notably Edmund Kunji in Cambridge, has done very elegant modeling to propose mechanisms for the action of the uncoupling protein. So now we need to move into physiology. And one of the classic things which was originally discovered by Hans Raphael very early on was that the nucleotide sensitive uncoupling, the strange thing about brown adipose tissue, was not constitutive, but it was inducible. And so at the moment of birth, there was a tremendous increase in GDP binding. And the GDP binding then decayed as the animals were grown and were reared in the warm. You could put them back into the cold again, and the GDP binding would reappear in parallel with the proton conductance. You can take this into the whole animal. This is an experiment by Jean Hims-Hagen many, many years ago. And you can infuse a whole rat with noradrenaline. And if the animals are warm acclimated, you show a small response. If they're in the cold, you see a very much larger response. So there is an adaptive induction of what we now call the uncoupling protein in the cold. And when we start looking at cells, so brown fat cells, we find that we can make cells from the cold adapted guinea pigs. We can add noradrenaline, and within a couple of minutes, we have a very high rate of respiration, which shows no respiratory control. In other words, noradrenaline, which is the physiological activator of brown adipose tissue in vivo, noradrenaline at the level of the isolated brown adipocyte can produce a tenfold or more than tenfold increase in the rate of respiration. When you inhibit the beta adrenergic receptor to which noradrenaline binds, respiration drops back to basal. Now that's a cold mitochondria from a cold adapted guinea pig. If you use the warm adapted guinea pig, which has very much less GDP binding, very much less GDP sensitive proton conductance for the mitochondria, the noradrenaline shows a very much diminished respiratory response to noradrenaline, respiratory response. So in other words, warm adapted guinea pigs, even though they show lipolysis from the noradrenaline, that noradrenaline does not activate the proton conductance pathway. With the cold adapted mitochondria or cells, you get a full activation of the protonophoric pathway. So if we compare the ability of fatty acids to uncouple brown fat mitochondria in the presence of nucleotide, we see something very interesting. We can take the cold adapted mitochondria, which contain this much uncoupling protein, and the respiratory increase as a function of free palmitate looks something like this. This is nanomolar free palmitate. This is palmitate buffers equilibrated with albumin. So we are way below 1 micromolar here. That's the cold adapted mitochondria. The warm adapted mitochondria, which have roughly 1/10 as much uncoupling protein, behave like this. And if you extrapolate to 0 uncoupling protein, you get this response there. So the uncoupling protein sensitizes the mitochondria to fatty acid uncoupling by a factor of about 30. There's a 30-fold decrease in the concentration of free fatty acids required to uncouple the cold adapted mitochondria relative to those lacking the uncoupling protein. We can ask a very important quantitative question. We can ask the question, how much fatty acid what concentration of fatty acids do you need to get those fatty acids oxidized? And so you can use coenzyme A, carnitine and ATP, that allows fatty acid activation oxidation, have an uncoupler to bypass the uncoupling protein. And now we're looking at substrate delivery. This is as though we were doing a seahorse experiment, looking at substrate delivery. And we find that as little as 12 nanomolar unbound fatty acid will give half maximal respiration. You need about 50 nanomolar free fatty acid for maximal respiration, as long as you've removed respiratory control. So that's fatty acid as substrate, extremely potent as a substrate. Fatty acid as a UCP activator, you use a non-uncoupling substrate, pyruvate, malate, and you don't have CoA and carnitine present, so the palmitate can't be oxidized. The only thing it can do is to bind to UCP and to increase its conductance. And there again, we find 100 nanomolar, very low concentration of palmitate is designed to activate, to increase release respiratory control. So basically, we have gone from a phenomenon of isn't it curious that brown fat mitochondria don't show respiratory control, to a very precise mechanism. We have the protein, we have the mechanism, and we have the evidence that fatty acids are both the substrates for thermogenesis and the activators of the uncoupling protein. So they play a dual role as substrate and UCP activator. And now, in the last few minutes or so, we'll finish a bit early with this, we can now develop a mechanism for the acute regulation of thermogenesis, which is beautifully self-regulating and has been challenged, but I think we've overcome the challenges that this is still accepted after nearly 40 years. Let's think what happens. The noradrenaline receptor on the plasma membrane, it's a beta-3 adrenergic receptor, and that gets activated by noradrenaline released by the sympathetic nervous system. And the noradrenaline receptor leads to the generation of cyclic AMP, protein kinase A, and lipoprotein lipase that hydrolyzes the triglyceride on the multilocular droplets, and the free fatty acid enters across the outer membrane through the porins. And in theory, the free fatty acids do two things: bind to UCP1 to activate it and act as substrate. So let's look at this now in a little bit more detail. And what we can do is to model this with isolated mitochondria. So I said that one of the things one tries to do is to get away from needing to work with isolated mitochondria and focusing on intact cells, but one can do experiments with isolated mitochondria that very closely mimic what would happen in the intact cell. And this is one of them, and this is really the experiment which explains the acute regulation of the uncoupling protein. We are measuring simultaneously current with an oxygen electrode and voltage with a tetraphenyl phosphonium electrode. So we're measuring current and voltage. And then we are mimicking the lipolysis by very slowly infusing fatty acid into the mitochondrial incubation, the stirred mitochondrial incubation. And as we infuse the fatty acid, we have CoA, carnitine, and ATP present so that the fatty acid can be activated and oxidized. As we do that, the fatty acid concentration, the free fatty acid concentration starts to rise. And that increase in free fatty acid concentration activates the uncoupling protein, with the result that the membrane potential starts to fall, and that allows the respiration to start increasing. And then at this stage, with the dotted line, we stop infusing the palmitate. This mimics the end of a thermogenic episode. And immediately the fatty acid concentration starts to fall because it's now continues to be activated and oxidized by the mitochondria. It's no longer being delivered. And the concentration of palmitate falls. And in parallel with that fall of palmitate concentration, the conductance decreases, the membrane repolarizes, and respiration goes back to a basal rate. And other intermediates like palmityl-CoA or palmityl-carnitine simply do not correlate with this increase in conductance. So to go through that cycle in detail, we start off with a non-thermogenic substrate, and this is mostly pyruvate in the case of the brown adipocyte. So pyruvate will enter, it will fuel the citric acid cycle, drive a conventional proton circuit with retained respiratory control. The first fatty acid that goes in cannot be oxidized at any particular rate because the mitochondria are showing respiratory control, which is inhibiting that oxidation. The fatty acid instead starts to accumulate and to bind to the uncoupling protein. It reaches the stage when the proton conductance is sufficient to allow rapid respiration. Acyl-CoA fatty acids are activated to acyl-CoA, acyl-carnitine, and oxidized at a high rate. This is the thermogenic rate of rapid oxidation, where the fatty acid is at the same time activating the uncoupling protein and acting as substrate. When we terminate the noradrenergic activation, lipolysis stops. The fatty acids are still there, but the affinity of the acyl-CoA synthetase is higher than that of the uncoupling protein. So it pulls the fatty acid off the uncoupling protein, and that recouples the cell, and one goes back to the basal situation. So, say a little self-indulgent going through these experiments, which completed some time ago, but I want to give some idea of the sort of thought processes that go on, how one gets from a vague phenomenon when one makes serendipitous discoveries. And Hans Raphael made the discovery of the nucleotide coupling. I found a slight bend in an oxygen electrode trace, which led to the chloride permeability. And then we worked through more or less conventional steps to determine what the uncoupling mechanism is doing and what it's caused by and how it's controlled. So I will stop at that stage. We have time for questions, general questions about proton conductance, uncoupling, brown fat, whatever you particularly like. That's the hero of the research.
I have a question. We've talked about proton conductance before, but not as much on coupling proteins.
Yeah.
And so it seems like there's a proton conductance that can occur in the absence of uncoupling proteins. Yes. And some of that is like ANT, phosphate carrier, you've talked about that. Is that all of it? Is it all protein mediated, I guess?
Martin has spent much more time looking at the endogenous proton leak than I have, and he believes in terms of correlations, it correlates to the amount of, for example, the adenucleotide translocator. Do you want to extend, Martin, on?
Yeah, so as David said in, I think, a previous lecture, there's a whole range of cycling processes like calcium cycling, which can net contribute to the proton leak. So we were interested in seeing if we remove that by removing all the cations, what's left. And as you keep passing the system by removing things, there's always something left. And in the end, we were able to show that something that's left correlates with the amount but not the activity of the adeноnucleotide carrier. So there is an adenonucleotide carrier uncoupling, partly by fatty acids moving through the ANT, partly through protons in some way moving through the ANT. But even if you block the carrier completely, then the proton conductance still depends on the amount of ANT there, which you can modify genetically, increase it or decrease it, and you find the proton leak follows that. So our model is that the presence of carriers like the ANT in the membrane just cause a discontinuity in the lipid bilayer, which allows protons occasionally to pass through the membrane, right, presumably by the side of the protein lipid interface. That's the model. And then when you have a conductance like that, Keith Garland showed quite a long time ago, then you get a non-ohmic response. That's the natural response of a conductance to a defect in the membrane, just from the thermodynamics of the way an ion crosses a membrane. So we think that's what gives you the underlying non-ohmic response of the endogenous proton leak. And as David said, it's correlational. It's hard to show directly, but I think it's a fairly convincing case. And then what biology seems to have done is imposed over that these uncoupling proteins to amplify it, but they also have this non-ohmic character. So you end up increasing the same phenomenon, but now under control of having a protein to do it.
What, do you have any sense of why there'd be specificity towards the ANT for that phenomenon?
Its amount. It's present in such enormous quantities relative to anything else apart from UCP and brown fat. I mean, they're all other of they're called what, SL a 25 class of membrane transporters. They're all present at least 100 times lower concentration than the ANT. So
yeah, so we did the same experiment that we did with the ANT with the UCP, again inhibiting it as far as we could tell completely, and then showing that the residual basal conductance, specifically in brown adipose tissue, also depends on the amount of UCP1. So exactly as David said, the model is that any member of this family in sufficient abundance will leak protons. That's actually disputed by some of the people who work on the UCP and say, no, we didn't fully inhibit the UCP. And it's always hard to know if you've done that or not. But our interpretation of it is, no, any mitochondrial six-membrane helix transporter will have this property. And it turns out to be quite useful because it means that if you have a mitochondrion that is very energy intensive, it has a lot of ANT to service that. It also has a relatively high native proton leak. And that's really good because it stops you going into dielectric breakdown. Because if you're energy intensive, not only do you have a lot of ANT, you have a lot of electron transport chain. And if you didn't have an endogenous leak, that could build up a much bigger PMF. And that, in the end, might cause dielectric breakdown of the membrane where you just electrically blast a hole in it. So we think it's a protective mechanism to prevent you from going to too high a PMF. That's why it's non-ohmic. So nature has taken advantage of this underlying property of ion leak.
So something which is very, very neat, if you like, is that the strongly non-ohmic relationship of the endogenous leak, which all mitochondria have, means that when you go from state 4 to state 3, when you're now generating ADP, there's a dramatic drop in the leak. So I used to use the analogy that it was like an old-fashioned steam engine sitting in the station, blowing off steam through its safety valve. And when it starts moving, the steam instead is used to drive the pistons, to drive the train. The pressure drops and the safety valve no longer blows off steam. So one can combine this protective mechanism that Martin talked about in state 4 under low respiring conditions with an efficient use of the proton circuit for ATP synthesis, where as a result of the lowering of the proton motive force. Other questions? PowerPoint?
Looks good.
Okay, fine. Right, so you're going to need two things for this problem solving. There's the brief PowerPoint which says students, which just contains the problems, and good idea to download that. And the other thing you need is the Excel program for the membrane potential calculations. So essentially, in the breakouts, I'm going to suggest manipulations of membrane potential, etc. And you're going to simulate them in the Excel spreadsheet. Discuss among yourselves what you think is happening, and then report back to the main session. So there'll be a series of relatively short 5-minute breakouts to discuss the actual problem. So everybody, I hope, has downloaded the students version, which is just 7 or 8 slides with the problems, and also have the Excel spreadsheet, which we're going to be dealing with.
Unable to download that. Can someone guide me where I can find this in the Dropbox?
In the Dropbox, let's have a look.
Yeah, David, I think it may be in the wrong folder. If you tell me the name of the file you want them to look at, I'll drop it in the Dropbox.
Okay, so
2020 P4 student problems.
that's it.
Okay, that's in the wrong folder. I'll just move it across now. So that's now in there.
Right, and the Excel spreadsheet, is that there as well?
So there's one under programs called Cell Stimulation Excel.
Yeah, that's it.
So that one's been there for a day or two.
Yeah, okay, fine.
Thank you.
Right, everybody found it now? Yep, okay.
So, sorry, is it the cell simulation sheet? Is
yes.
That right? Yeah.
Yeah.
Cool.
And essentially, we're going to be playing with that and interpreting the results a little later. Okay, so necessity is the mother of invention. And some 20 years ago, I started looking at or trying to investigate mitochondrial membrane potential in cultured neurons, and using lipophilic cations, which were sold as mitochondrial membrane potential indicators, and getting results I simply did not understand. And then looking in the literature, other people were getting results they didn't understand. And I realized one had to go back to first principles to try to understand how these probes work, how they respond to changes in mitochondrial membrane potential, plasma membrane potential, etc. And that led to our development of that Excel program, which is surprisingly accurate in predicting responses. It has 2 roles. First of all, you can plan your experiment and then see what effect a given change in membrane potential would have on the fluorescent signal. And equally importantly, you can interpret experiments. So you can do an experiment and then you can try to fit the simulation to the experiment to get a semi-quantitative idea of what was happening during that particular edition. And we'll be dealing with examples of both of this. So today is the voltmeter. You've had the ammeter, and now we're having the voltmeter. And the technique which Martin and I, Akosh, other people have been using to measure membrane potential relies on lipophilic cations being permeable across the inner mitochondrial membrane and not being pumped, just being passively permeable and coming to equilibrium. And if you have an electrode, you can use, for example, tetraphenylphosphonium and measure the residual external concentration. In the early days, we used radioisotopes, and generally people now use fluorescent probes. And these have rather special properties. That the 2 we're going to talk about are tetramethyl rhodamine methyl ester, and the other is rhodamine 1,2,3. And I briefly mentioned this in the first lecture, that unusually this charged species is permeable across the plasma membrane. And it's permeable because that positive charge, the lack of an electron, is delocalized over the entire structure with alternating single and double bonds, which is called a pi orbital system. And that produces sort of a cloud of very low density cloud of charge, which doesn't prevent it from crossing through a lipid bilayer. Very closely related to tetramethyl rhodamine ester is rhodamine 1,2,3. You can see the only difference is that the nitrogens, instead of having two methyl groups, have two hydrogens. And an important factor for permeation is that these compounds should be as hydrophobic as possible. And so the clear difference is that these have additional methyl groups and these have hydrogens. And so the rhodamine 1,2,3 is less hydrophobic than TMRM. Otherwise, they behave very similarly. And what this means is that rhodamine 1,2,3 takes about 20 times longer to equilibrate across the mitochondrial membrane than does TMRM. And this is really important because it's going to affect how we design experiments and how we, for example, do the loading and the equilibration. So how do we actually do this? What we're dealing with is single cell fluorescence. So here's a high resolution image actually of one of our insulin secreting cells. And you can see the individual mitochondria there. So in theory, you could estimate the mitochondrial membrane potential by measuring the fluorescent gradient between the mitochondria and the mitochondria pore region, the cytoplasm. In practice, it's extremely difficult because you're trying to work with the roughly 300-fold different in intensity, and the errors in sort of background correction are really too large for any form of quantitation. What we do instead is we measure the total fluorescence of the cell, and then we estimate how much that fluorescence is due to the cytoplasm, the residual TMRM, for example, in the cytoplasm, how much is due to the TMRM accumulated into the mitochondria. So this is really restating what I've just said. No probes are perfect. Some of the least bad are TMRM and rhodamine 1,2,3. And TMRM takes about an hour to equilibrate across the cell membrane. And rhodamine 1,2,3, you normally load it for a brief period at a very high concentration. I mean, we're talking in low nanomolar concentrations for TMRM, for example. So with rhodamine 1,2,3, we might use 1 micromolar for 20 minutes or something like that to push it in a certain amount, and then we wash to get rid of it. If any of you are using these fluorescent techniques, you might have come across or read about something called JC1. JC1 is pretty, but pretty useless. It gives a red fluorescence when it's aggregated and a green fluorescence when it's monomeric. And crudely, that is said to be a way of monitoring the membrane potential. So red mitochondria are polarized, but it's pretty useless. It aggregates, it precipitates, and generally it should not be used. I'm going to talk about one or two other fluorescent probes more in a sort of a warning basis, either in this lecture or in the final lecture. So in order to understand how these probes work, you simply have to understand 3 basic principles. The 3 principles are actually used to generate the Excel program that you're going to use. The first, probes reach Nernstian equilibria across both the plasma membrane and the inner mitochondrial membrane. The Nernst equilibrium essentially says for each 60 millivolts, there's a 10-fold equilibration at equilibrium. It's a logarithmic relationship, so 120 millivolts means 100-fold equilibration. If you have 180 millivolts, it would be 1,000-fold equilibration. That's the first one. And an absolutely critical feature of all of these fluorescent probes is that they should come to a passive equilibrium across the membrane. If there's anything that's trying to pump the probes either in or out, the technique is invalid. And you sometimes find with cell lines that cell lines have developed multi-drug resistant transporters that take a dislike to things like TMRM and try to expel it. In fact, there are some studies use TMRM to detect whether there is a multi-drug transporter, which is really basically screwing up the equilibrium. And if it is, you either have to use a different cell line or you have to inhibit that transporter to make sure you have a true thermodynamic equilibrium and it's not being pumped. Second point is we are dealing with the transport across two membranes, across the inner mitochondrial membrane and across the plasma membrane. And as I've tried to show in this figure, the surface to volume ratio of the mitochondrial matrix in a membrane is thousands of times greater than the surface to volume ratio across the plasma membrane. And this is simply due to geometry. So the probes don't really know whether it's crossing a plasma membrane or a mitochondrial membrane, but the equilibration is entirely different. And so, for example, equilibration across the plasma membrane with TMRM could take 30 minutes to an hour, whereas equilibration between the matrix and the cytosol, we assume it's complete within the time resolution of our experiment, so it's virtually instantaneous. The third principle, which is absolutely critical, is above a critical concentration, the probes aggregate, and the aggregated probes become non-fluorescent. So if you start loading up the matrix with TMRM, the fluorescence increases up to a level, and then it doesn't increase anymore, even though more TMRM is going into the matrix. That additional TMRM is aggregated and invisible, it's non-fluorescent. When you release the probe from the matrix back to the cytosol, it immediately becomes fluorescent. So you're going to see a rather curious business that the release of probe from the mitochondrial matrix under some conditions gives an increase in whole cell fluorescence, because aggregated non-fluorescent probe in the matrix comes out and becomes monomeric fluorescent probe in the cytoplasm. And understanding and appreciating the conditions where you have this aggregation is absolutely critical to understanding the experiments, both to design and interpret your own experiments and to interpret the experiments which are in the published literature. So the Nernst equation, basically, as I say, I'm very sloppy about whether it's 59, 60, 61. I don't think it matters too much. The distribution between the mitochondrial matrix and the cytosol is governed by the mitochondrial membrane potential. It's very rapid. The distribution equilibration between the cytosol and the external medium is governed by the plasma membrane potential, and that is very much slower. So if we look at a cell with a plasma membrane and a mitochondrial membrane, and we say maybe it's a neuron with 80 millivolts of membrane potential across the plasma membrane, 150 across the mitochondrial membrane. Electrophysiologists normally give this a negative sign. We don't really bother with that, I'm afraid. It means from the Nernst equilibrium, if you could start with 10 nanomolar of something like TMRM outside, the 80 millivolts will give it a 30-fold accumulation to the cytoplasm. So it could be 300 nanomolar in the cytoplasm. The 150 millivolts across the mitochondrial membrane could shoot that up to 90 micromolar in the mitochondrial matrix. So the overall accumulation is enormous from external medium to matrix. And that's one of the reasons why we use very low nanomolar concentrations. There are some probes you've got to go below 1 nanomolar to avoid artifacts. So what we're going to do now is to manipulate this program. This was a program we set up for a paper we published way back in 2000, actually, in order to understand our puzzling fluorescence traces we were getting with our cultured neurons. So I want to talk my way through this. This isn't the actual program, this is just a screenshot of the program. And we have a number of variables. We assume that in this particular case, the mitochondrial volume is 4% of the total cell volume. So the mitochondrial matrix occupies 4% of the cell, and the cytosol occupies the other 96%. Now, that is tunable, but we're going to keep it at that value for this particular problem solving. The probe concentration we're going to vary. So we're going to start off with a default of 25 nanomolar. And we're going to use two probes. We're going to use TMRM, and we're going to use rhodamine 123. And as I said, TMRM equilibrates more rapidly than rhodamine 123, about 20 times faster. This is the rate constant for equilibration across the plasma membrane. So for TMRM, it's 0.04. For rhodamine 123, it's 0.002. Now, the next variable we have is the quench limit. That's the concentration of probe in the matrix at which aggregation just begins. And again, we've set that here to 30 micromolar, just for the example of these experiments. And then we're running experiment, and we can alter the time scale simply. So we're taking individual time points at every 10 seconds, and we have about 100 or 200 time points, which I'm not actually showing. They're hidden beneath this spreadsheet down here. So we can set up a protocol, and I've set up these various time interval protocols based on 10 seconds that we're going to, at 90 seconds, we start off with basic conditions, then we can change plasma and mitochondrial membrane potentials at that time point, we can change them at that time point, and we can change them at that time point as well. If you get confused and you think you forgot where you started with, these are the default values we go back to when we want to reset the experiments. The default plasma membrane potentials, 80 millivolts. My program needs a minus sign there, but don't worry about that. The default mitochondrial membrane potential is 150 millivolts there. So that's the setup. Now, what do we have over here is the readout. And the readout, the y-axis automatically adjusts. It gives you the total cellular signal, the signal from the mitochondria, and the signal from the cytoplasm. So what we are measuring is the total signal, which is the sum of the cytoplasmic signal here and the mitochondrial signal here. And we're going to see how that varies with time. Everybody happy at that stage? Everybody raring to go to play with this wonderful program? I hope so. Okay, open the cell simulation. Stay looking at this PowerPoint. Don't break out yet. So your simulation, as I've said, will look like this. If you can sort of keep the Excel program in the background for the moment, so you're looking at my PowerPoint. Total fluorescence in blue, mitochondrial fluorescence in orange, and cytosolic fluorescence in, I forgot what color that was originally, but it sort of washed out in taking the screenshot. Okay, I've described most of this already. It's assuming that equilibration across the mitochondrial membrane is instantaneous. It's within the time course of the experiment. The plasma membrane is much slower, and we're using rate constants of 0.04 for TMRM, 0.002 for rhodamine 123. And I'm not a computer expert. It's a very clunky program which uses repetitive iterations to approach equilibrium, but it works. So quench mode, we've set at 30 micromolar, and the probe fluoresces when it's released into the cytoplasm. The default settings will put you in quench mode, basically. You assume the matrix is 4% of the total cell volume, and the graphs show the 3 separate parameters, total, mitochondrial, and cytosolic. And anything on the program that is in blue is tunable. Anything that isn't in blue is fixed. Everybody okay so far? First thing we're going to do is we're going to add a bucket of FCCP. We're going to depolarize the mitochondria in these cells. We're going to be fairly careful. It's not going to affect the plasma membrane. I told you earlier that too much FCCP collapses the plasma membrane, but we'll keep the membrane potential at the same. So what we're going to do is to change the mitochondrial membrane potential at number 3. We're going to change this to 0. And then that's the first part of the exercise. The second part of the exercise is that we're going to change from TMRM to rhodamine 1,2,3. So the exercise, when we're going to break out in a few seconds, is going to ask you to simulate a collapse of mitochondrial membrane potential in an experiment using TMRM and an experiment using rhodamine 1,2,3. And to do that will just take you a few seconds. And then what you've got to do is to discuss what is happening and try to understand what is happening. And then we'll resume the main session. And I think I said 5 minutes. We might need less than 5 minutes, but at the beginning we'll give it 5 minutes. So everybody is ready now. You've got the program loaded. You know what you're going to do with the mitochondrial membrane potential here and the rate constants. And let's go. So let's have a 5-minute breakout. Ricky.
Okay, here we go.
Okay, is everybody back?
Everybody's back.
Right. Welcome back. Let's talk about exercise 1. Now, you will all have got the same result, so it's not no prizes for getting the result, but we're going to discuss exactly what it means, and I want some input into this. So that's the response that you got from suddenly collapsing the mitochondrial membrane potential. And I need somebody from one of the groups to explain what they think is happening here. And I can't see all of you. I can just see a small selection, so I can't point to people. But
you can volunteer breakout groups by number if you want.
Mm-hmm.
Okay, let's do this. Breakout group 8. Let's have something from breakout group 8 to explain this extraordinary result. Breakout group 8 there?
Yes.
Okay. So you don't need to know the answer, but just say what you think is going on here.
So when the FCCP is added,
yep,
mitochondrial signal went down, all the way down, because there's no membrane potential. Now it's all compromised. But the cytosol.
So yeah.
Yes.
So where's the TMRM gone?
So it's left the mitochondria and diffused out into the cytosol.
Okay, and why do you get this tremendous peak?
I postulated that the spike is because the dye is now diffused across a larger area, so the fluorescence intensity appears to be larger even though the dye concentration is the same?
No, it's more subtle than that. It's dequenched.
Hmm.
So all of that excess TMRM was invisible in the matrix because it was quenched as an aggregate. When you release it to the cytoplasm, it becomes fully fluorescent. So what you're seeing is the dequenching of the probe as it comes out of the matrix into the cytoplasm. Is that okay? But the question is, where does it go?
So eventually it should diffuse out of the plasma membrane because the potential is different.
Yep. So that it's leaving the cell. It's crossing the plasma membrane. It actually it always surprises me it's so quick. That when you've released this large amount of TMRM into the cytosol, you've greatly disturbed the equilibrium distribution between the cytosol and the outside. You've got 10 times as much TMRM in the cytosol than you would have at equilibrium. So the excess leaves the cell quite simply and comes out to restore equilibrium. So you get this spike and recovery. The spike is the dequenching as it comes out to the cytosol, and then the subsequent fall is the probe leaving the cell, crossing the plasma membrane. So the next experiment was doing the same thing with rhodamine 123. Somebody else, group 7? Why are these two traces so different? Anybody in group 7? Hello, are you receiving me?
Yeah, because rhodamine is slower to reach equilibrium due to its membrane permeability issues.
Yep. We've already said it permeates 20 times more slowly. I've kept the other parameters constant, so you get the same spectacular spike of dequenching as it comes out into the cytosol. And then it leaves the cell, but it's much less permeable across the plasma membrane, so it takes much longer to re-equilibrate. And even after, these are seconds, even after 30 minutes, it still hasn't fully re-equilibrated. So what we've got from this first exercise is the understanding if you collapse the mitochondrial membrane potential and you are in quench mode, and by quench mode, you mean you've loaded with a sufficient concentration for the probe to aggregate in the mitochondrial matrix. When you collapse the mitochondrial membrane potential in quench mode, you get this spike increase of fluorescence. It's really exciting to actually see this looking through the microscope. And then it decays. And it decays more rapidly with TMRM and more slowly with rhodamine 123. Everybody happy with this first experiment? Yeah.
Is that just diffusion, or is there an active?
No, it's pure diffusion, pure approach to an Ernst equilibrium. If there's any active pumping, the technique, the probe is invalid because it has to be a natural distribution due to the Ernst equilibrium, and nothing is being pumped anywhere. It's purely responses to the membrane potentials. Okay.
You probably answered the question, but why does one diffuse so much quicker than the other? I'm sure you said that before and I missed it.
Okay, not at all, no. This is interesting. I took this today from the original paper we published, Manus Ward and I, in 2000. And this was an actual experiment with potassium chloride to depolarize the plasma membrane of a neuron and FCCP. And this was the actual experiment we did with the addition of FCCP, the fluorescence. And that was with rhodamine 123, and this was with TMRM. So that's an actual spike we got with TMRM. And putting into the program a collapse of the mitochondrial membrane potential with FCCP and a plasma membrane depolarization with KCl, the program produced this trace. And you see how similar the program trace is to the experimental trace. And again, this is the experimental trace, the real experiment. This is the simulation. So essentially, we were altering the parameters to get the simulation to look like the experimental trace, and then reading off those parameters. What was the change in membrane potential? What was the mitochondrial volume, etc. Okay? Now, the most important message you're going to take away today is the importance of knowing whether you're in quench mode or non-quench mode. And the criterion of quench mode is when you collapse the membrane potential with FCCP, you get one of these spikes. And so what I want you to do for the second problem is to keep the mitochondrial membrane potential collapsed here, which is number 3, keep the membrane potential at 0. We're going to work with TMRM, so set it to TMRM, 0.04 for the rate constant, and then progressively reduce the probe concentration from 25 nanomolar. And I want you to do this, and you can sort of check. It might be an idea if just one person does it and you share the screen with the other 3 or 4, and just work out at what stage you lose quench mode. When do you drop below the level that results in aggregation of TMRM? And okay, let's do that for, so this is exercise 2. Reducing the concentration until you lose the quench spike. Okay, breakout, please.
Looks good.
Yep, and a subtle depolarization of the mitochondrial membrane potential in quench mode. And let's work our way back through the breakout groups. Let's go back to group 5 and ask group 5 to talk a little bit about this.
So I think the main conclusions we drew were if you were looking at before and after, like you said, your 2 different scenarios were one you had the decrease and one you didn't, you wouldn't see a difference between the 2. The signal would be the same.
So if you, in other words, if you did this outside the fluorescence imager and then put the final result into the fluorescent imager,
it would look exactly the same.
It would look exactly the same. In other words, you would get no information about the change in mitochondrial membrane potential.
Yes.
Yeah. This is extremely important point, and this probably is the most important take-home point, that if you're working in quench mode, the changes are transient, and you detect them by monitoring the experiment when you make the addition. If you want to do fluorescence activated cell sorting, for example, where you have 2 sets of cells and one cell you've knocked out a component and that's dropped the membrane potential, and you load them in quench mode and you put them into the FACS scanner, you'll not see anything. It is incredibly common, especially in the early days of these experiments, for people to work in quench mode with FACS scanning without understanding what they're doing. There's a probe which was very popular early on, which is called Diox-6. And Diox-6 has an extremely low quench limit. You have to work less than 0.5 nanomolar to get out of quench mode. Anything above 0.5 nanomolar, you find it stops responding to mitochondrial membrane potential if you were doing a FACS experiment. So if you were trying to detect a difference in mitochondrial membrane potential, you wouldn't see it. What you will see, to add to the complication, is a difference in plasma membrane potential. So if you find yourself looking at a FACS experiment and it's been done in quench mode, then they're looking at the wrong potential. And there's some very embarrassing situations which I might mention in my final sort of rant lecture after this, where people have actually done this. So absolutely critical. Are they in quench mode? Are they in non-quench mode? If they're in quench mode, ignore the results that they claim getting from FACS scan. Everybody happy with that, or unhappy? I hope I haven't destroyed anybody's research careers by actually saying that, but it's extremely common. It was, I mean, we discovered this in 2000 when we did made this program and did a simulation. A year before, there's a very good Israeli bioenergist called Haggai Rottenberg, and he actually experimentally demonstrated that you lose the mitochondrial sensitivity in quench mode when you're doing a FACS scan. He did a very sort of elegant series of experiments just the same time that we did our simulation, and we came to exactly the same conclusion. And early experiments using diox-6, 10 or 100 times higher concentration, were not detecting changes in mitochondrial membrane potential, detecting changes in plasma membrane potential, and that rather alters your interpretation of your experiment, I'm afraid. So we've really answered this question down here. If you're doing FACS scanning, you have to be in non-quench mode. It is absolutely essential. And I think we've done this. We actually skip over this. We know how to design a FACS experiment. We need to go below quench mode. So let's go back here and let's break out and go into non-quench mode and see how that trace works. I can do it here. There's no point in going out. This is what would happen if we kept the same experiment, 120 millivolts, that drop in potential. We've now gone from quench mode, which was 25 nanomolar TMRM. We've gone to 4 nanomolar TMRM. And this is what we are getting. We're getting a permanent change in whole cell fluorescence. We're releasing some of the TMRM, not all of it, because the potential is only dropped by 30 millivolts from 150 to 120. There is a brief hiccup of increased cytosolic fluorescence, which then rapidly comes back to baseline. So we have a permanent difference. So in other words, if you were comparing your wild type and knockout cells in non-quench mode, and these are the control cells, and these are the knockout cells, you will detect a difference in whole cell fluorescence. So non-quench mode is absolutely essential. Okay, I think we've hammered that home pretty well. I do it in capitals here as though I'm tweeting from the White House. You must not use FACS in quench mode. So it's essential, first of all, when you're designing an experiment, to establish whether you're in quench or non-quench mode. If you're interpreting somebody else's experiment in the literature, are they in quench or in non-quench mode?
I have a question.
Yes, please.
Is this cell dependent, the concentration? Like, would we have to optimize the TMRM concentration?
So this is not an experiment, it is a simulation. So I have put in expected values of plasma membrane potential, mitochondrial membrane potential, the quench limit, the concentration of probe, all of those, I've sort of tuned them for the neurons I was working on when I was doing these experiments some years ago. So each cell is going to be different. So it is not, I mean, Akos has some very elegant techniques where he can get absolute numbers out for his mitochondrial membrane potentials. This does not give absolute numbers. It gives a sort of semi-quantitative, semi-qualitative estimate of what the trace will look like. But if you're using, for example, a liver cell, you would tune down the plasma membrane potential from 80 millivolts to maybe 40 millivolts or so on. If you had a brown adipocyte which has a mass of mitochondria, you might increase the mitochondrial volume above 4% up to 10% or 20%. And so essentially it's a way, it's not a way of getting an answer, it's a way of thinking about what sort of variables would give the experimental result we're actually seeing. So it's not, so it has that limitation. It's not truly quantitative.
But the other way to answer the question is, if you're working in some different cell type, you need, and you're working anywhere near the quench limit, you just need to do the experiments to find out whether you're above it or below it.
Yeah.
But in practice, like 4 or 5 nanomolar, that in most cells, like neuronal cells, cardiomyocytes, that was in non-quench mode, and in a liver cell that was in other end of the palette, 20, 30 nanomolar could be in non-quench mode. So that's the practical.
So the FCCP test would be a good test, kind of a thing.
You need to do every time, every cell type. Yes.
Right.
I have a question.
Yeah.
So theoretically, when we use brown adipose cells, assuming they are UCP1 activated, TMRM will not give you a potential, right? Unless you are GDP or anything.
Well, if we're talking intact cells, the intact cells, brown adipocytes, always have their nucleotide in the cytosol. The cytosol is full of ATP and GDP. So the physiological transient in a adipocyte is plus or minus fatty acid. The nucleotide is already there. And I did the brown fat work 20 years more or less, or 20 years before I did the membrane potential work on the neurons, and I never went back to do the membrane potential experiments on the adipocytes. Maybe one day I will, I don't know. Okay. Now there's a new exercise, which is called exercise 4, which is here. And we're going to put all the defaults back. We'll actually keep with rhodamine 1, 2, 3 up here. And we're going to stay in non-quench mode. So 4 nanomolar, rhodamine 1, 2, 3, 4 nanomolar. And then we've repolarized the plasma membrane potential. Now we're going to depolarize the we repolarized the mitochondrial membrane potential. We're now going to depolarize the plasma membrane potential, and we'll do it for 1, 2, and 3. So we're going to take the plasma membrane potential from -80 to -30 for 1, 2, and 3. And you'll do this in the breakout, and then you'll talk a bit about it, because it might strike some sort of bell in terms of the experiment we've just done with changing the mitochondrial membrane potential. So let's do that first. Sorry, first of all, we do it with rhodamine 1, 2, 3, and then we do it with TMRM. So the same experiment, first with rhodamine 1, 2, 3, and then with TMRM, simply depolarizing the plasma membrane potential from -80 to -30 millivolts. So let's break out, folks, and come back in 5 minutes. We started with rhodamine 1, 2, 3, and we depolarized the plasma membrane, and not very much happened. We got a slow loss of fluorescence across from the cell, from both the mitochondrion and from the cytoplasm. So we've depolarized the cytoplasm, so we're going to lose probe from the cytoplasm. And that means the concentration of the cytoplasm is less. So even though we're doing nothing to the mitochondria is going to lose the signal, because it's trying to keep the same gradient between matrix and cytosol. And the cytosol concentration is decreasing. So I'm just getting this slow fall. If we change to the much more permeant TMRM, we get this trace. And the question I asked one of the groups I popped into was, whether this reminds you of anything. And let's go back. How far have I got back? Group 5, for example. Group 4. Let's have group 4. Can somebody from group 4 tell me if this time course, if they've come across this time course, this sort of time course a bit earlier in the problem solving? Group 4. Anybody there?
That would be PK, Mustafa, Lily, Durai, and Cher. Sure.
Okay. Are you receiving me?
Nobody wants to.
Yeah. Yes, we are.
Okay. Where have you seen this before? This type of trace?
Yeah, sadly, I don't have any idea. But if someone else in my group can express, that would be great.
When we depolarize the mitochondrial membrane potential in non-quench mode, by the similar effect, we dropped, remember, the mitochondrial membrane potential from 150 to 120 millivolts. We got a trace that looked almost identical. I don't have my program loaded. I could load my program actually. Just show you. No, I don't know how to do that. Okay. Here, so do you see the?
Yeah, we see that. So you're saying it was it's like shorter. It was a quicker drop. It was because the TMRM it was being more permeant.
Yeah. Okay. So yeah, fine. So let's go back to an earlier experiment. Let's remember that one. Let's go back to repolarize, and we'll depolarize the mitochondrial membrane. And in non-quench mode, I think it's probably better to do it with rhodamine 123. And we'll actually just do a bit of fiddling here. Move that one out. Move this out of the way. What I'm trying to show here is that this is the rough shape of a mitochondrial depolarization in non-quench mode. And what you've just been looking at is a plasma membrane depolarization in non-quench mode, which looked more or less the same. So the point I'm trying to make is we have an ambiguity here. The ambiguity is that how do you know which membrane you're depolarizing, whether it's the mitochondrial membrane or the plasma membrane in non-quench mode? And the answer is you very frequently don't unless you have very clear ideas of the sort of experiment you're doing. So one of the problems with non-quench mode is that it does not distinguish between changes in plasma and mitochondrial membrane potential. And so these bottles of TMRM that are sold at enormous expense as mitochondrial membrane potential indicators are also plasma membrane potential indicators, and this is a distinct problem. So the point I want to make here is the ambiguity. So that was ambiguous. And the question is, how do you overcome this ambiguity? So yeah, I should have just shown that this is a plasma membrane depolarization. This is a mitochondrial membrane depolarization. The kinetics can be different, but the extent is essentially the same. So we have an ambiguity. What do we do about it? In non-quench mode, it's difficult to distinguish between the two. And so what we can do, and which I started, which Akosh has greatly refined, is to include a second ion, to include a lipophilic anion together with a lipophilic cation. And the lipophilic cation, TMRM, we've already seen how that's accumulated into the cytoplasm, into the matrix. The lipophilic anion, and we use this from a molecular dynamics kit, and we call it the plasma membrane potential indicator, has the inverse relationship. It's expelled by a polarized plasma membrane potential and has a snowball's chance in hell of getting into the mitochondrial matrix with its high membrane potential. So if one can simultaneously measure the distribution of TMRM and PUMPy in non-quench mode, then you can estimate the plasma and mitochondrial membrane potentials. And Akosh has refined this greatly into a really sort of quantitative analysis. This is showing what happens when we depolarize the plasma membrane potential, going from increasing the potassium concentration. The PUMPy fluorescence gets brighter and the mitochondrial fluorescence gets dimmer. And we can put this into various equations. This is going back to NMDA and excitotoxicity from yesterday. The fluorescence increased when we depolarized with NMDA, repolarized with MK801, which is switching off the NMDA receptor. And we go through our simulation, and it's there are lots of degrees of freedom in this simulation, but you get this sort of response going from -80, you know, the MK801, you go back again and you depolarize with KCl. One interesting thing which came up from doing these experiments was that you only have to go up slightly in terms of FCCB concentration. A low FCCB concentration is selective for the mitochondrial membrane potential. You only have to go up tenfold, and the plasma membrane potential depolarizes as well. So experiments with intact cells using more than the absolute minimum amount of FCCP can be ambiguous because you can be collapsing the plasma membrane potential as well as the mitochondrial membrane potential. This is Akosh's paper where this is put on a quantitative basis there. There he is. So we now want to detect rapid changes in mitochondrial membrane potential doing experiment with rhodamine 123 as a probe. And I'll explain the reason that we're doing this in a minute. So we're with rhodamine 123, probe is back to 25 nanomolar, quench mode, and we've just dropped the mitochondrial membrane potential by 10 millivolts. And when we do this, we can actually show what happens. You don't need to go on to a breakout in order to show this. Let me have a look, see what I'm doing. No, I know what I wanted to do. Okay, I'm sorry. So with rhodamine 123 in quench mode, I want you to do two experiments. In one, I want you to drop the membrane potential from 150 to 140. And in the other, I want you to raise the membrane potential from 150 to 160 millivolts. That's the exercise. Okay, so just two very simple things. And then I want you to imagine conditions in which the membrane potential could change by this small amount, and what information you could get from looking at a cell population. So let's get this clear. I'm getting myself a little bit confused. I only did this yesterday. So let's assume that the slight hyperpolarization was caused by oligomycin. And then we ask the question, is it best to work in quench or non-quench mode? And why would oligomycin hyperpolarize the mitochondria? And under which circumstances would oligomycin depolarize the mitochondria? So these are the questions you're going to have a bit of freedom here to play about, and we'll give you 10 minutes for this, with changing the mitochondrial membrane potential from 150 to 140, or from 150 to 160, trying it in quench mode and non-quench mode, and then trying to discuss what the physiological relevance of this, what sort of conditions would result in oligomycin causing slight increase in membrane potential or a slight decrease in mitochondrial membrane potential. Now I've got it right. Now go into your breakouts. And this is the actual live program here, I was asking what happens when you subtly change the mitochondrial membrane potential. Take the membrane, slightly depolarize, minus 150 to minus 140. We hope we get it right. And we see a transient increase in the cytosol. It comes back, no change in the mitochondrial signal. So we get a temporary increase in fluorescence as a result of the mitochondrial depolarization. If we keep it depolarized and we go to rhodamine 123, the effect is much more long-lasting. So, but the transients are rapid, and the point is, quench mode is a very sensitive way of detecting rapid changes, even quite small changes during the experiment. Now, if we went into non-quench mode, with rhodamine 123, we'd see essentially nothing. We'd have to wait for hours and hours, and we wouldn't detect any real information about that rapid change in mitochondrial membrane potential. So let's go back to quench mode again, and we'll stick with rhodamine 123, and now let's slightly hyperpolarize to minus 160. We get this response. So we're very sensitively detecting the slight mitochondrial hyperpolarization by the addition of oligomycin. So the question I asked, and I mentioned this very briefly in passing yesterday when I was talking about glutamatic cytotoxicity, was under what circumstances would oligomycin hyperpolarize a cell, and under what circumstances would oligomycin slightly depolarize a cell? And I don't know what we got back to in groups. Let's go back to group 3 and see if group 3 could have any idea about the different conditions where oligomycin will go in opposite directions. Anybody from breakout group 3 likely to contribute to this? Is there anybody there on the other
sure,
side?
yeah, I can go for the group.
Yeah.
So we said ATP synthase will hyperpolarize when it's doing ATP hydrolysis and consuming ATP.
Let's get this the right way round. Okay. You're on the right track. Let's get it. Let's get it the right way around. What condition will oligomycin hyperpolarize the mitochondria? Yeah. Let's say you've got good cells and damaged cells. Which cells will hyperpolarize with oligomycin? The good cells or the damaged cells? Anybody?
The
Good cells.
Yeah, the good cells will, because you're decreasing the proton current, inward proton current through the ATP synthase. Now, if you have a damaged cell or partially inhibited electron transport chain, and this is a real cell, so the cytosol is producing glycolytic ATP, that ATP can reverse the ATP synthase. And so it becomes an ATP hydrolyzing proton pump, trying to keep the proton motive force up, to keep the membrane potential up. So this is quite a neat experiment. I rushed through this a bit yesterday with the excitotoxicity, and I showed you that you could look at a field of cells and work out how many cells are sufficiently healthy to be generating ATP, at least until you added the oligomycin, and how many cells were damaged and only keeping a membrane potential by hydrolyzing glycolytic ATP. This is the thing we call the oligomycin null point experiment. And you can actually look at a population. So you can look at 100 cells, a field of 100 cells, and say 65 of those cells are healthy and generating ATP, and the other 35 are damaged and hydrolyzing ATP. So you can quantify the extent of damage without having to measure the membrane potential. You're just asking, is the membrane potential sufficient for ATP synthesis, or the proton motive force rather, or is the proton motive force insufficient for ATP synthesis? Okay, I think that was the information I wanted to give there. And the other thing is, if you're dealing with a rapid transient, it's very difficult in non-quench mode to resolve it, because things are just so terribly slow. 4 nanomolar here. You know, it takes forever, basically, compared with quench mode. Right, I want to skip over a few things now. I want to go forward quite a few slides and just ask one question. And I'll answer it actually, because we're getting towards the end of this lecture. If you want to improve the sensitivity of an experiment in quench mode, you want to check the changes in delta psi m mitochondrial membrane potential, and you had sufficient resolution, would you look at the whole cell, the cytosolic, or the matrix fluorescence? I think you know the answers already from the experiments you've done. You look at the matrix. And so this is an actual experiment we did some time ago here in Lund. And
we're still on your previous screen, David.
Stop share, it says. Okay, let me get this right. I get very confused here. So let's share this. Okay, are you seeing improving sensitivity now?
No, we're seeing you.
Better.
What is it? Are you seeing a slide?
Yes, now we see improvement.
Yeah, okay, yeah, I will eventually learn how to use this wretched Zoom business, but thanks for warning me. So the question is, if you want to improve the sensitivity, what do you look at? And the answer is you look at the mitochondria pore regions in quench mode. And what you're measuring when the mitochondria hyperpolarize is the decrease in fluorescence in the non-mitochondrial region. This is actually the nuclear region appears to equilibrate fully with the cytoplasm with regard to TMRM. And so you can measure really very subtle mitochondrial hyperpolarizations. These are the effects of low glucose concentrations or pyruvate concentrations on the mitochondrial membrane potential of an insulin secreting cell, an 83213 cell. And you can follow the kinetics of the mitochondrial hyperpolarization with different glucose concentrations by really just looking at the relatively small non-mitochondrial signal rather than this large constant background of mitochondrial signal there. You need to have quite a good resolution microscope to do it. So a cell is not simply a blob, that you have sufficient structure and are sufficiently confocal that you can basically exclude the mitochondria from the field of view. These 83213 cells, it works rather well with. And I think we've covered this, which is the questions when that oligomycin hyperpolarizes healthy mitochondria and oligomycin depolarizes damaged mitochondria in an intact cell. And so you can do a population study. I think what I want to do finally is to go through an experiment, which was the experiment that started all of this going for us 20 years ago, which is back to our intact neuron and the NMDA receptor. And what we wanted to do was to monitor the changes in the two potentials under these sequence of events. And essentially, using exactly the protocols we've talked about with TMRM, the actual experiment of adding glutamate at this stage to activate the NMDA receptors, adding oligomycin to check for the oligomycin null point, and adding FCCP to check for quench conditions. This was the actual experiment. And by setting the parameters to the following, that the NMDA depolarized the plasma membrane potential from -80 to -30 millivolts, go back one, and the increased ATP demand and the calcium uptake depolarized the mitochondrial membrane potential from 150 to 145 millivolts. That produces this little upward spike here, which you also see there. The addition of oligomycin in this particular cell hyperpolarized, which showed that this cell was still healthy and generating ATP. We did this same experiment with an entire field of 100 cells and worked out how many cells were still producing ATP, how many cells were damaged and depolarizing with oligomycin. And that shows you the tightness of the fit between the computer simulation and the actual experiment. And that was with TMRM. And then we did the same experiment with rhodamine 123. We simply altered the rate constant for to change to rhodamine 123. This is the actual experiment, the upward tick with glutamate, the oligomycin showing that that cell is healthy, and the FCCP dequenching. And again, we can more or less precisely simulate that using rhodamine 123. So I think we have come to the end of this. It is complicated, but I think what have we learned today? We've learned the basis of what causes the fluorescence changes. We've learned that it's complicated. It's absolutely vital to know whether you're in quench or non-quench. That if you're reading a paper using rhodamine 123 or TMRM, you get very different changes. That you can be ambiguous, you don't know whether you're changing the mitochondrial or the plasma membrane potential, and that there are ways of overcoming this. So I think we'll stop at that stage. It is 8 minutes past by my clock. We can answer questions. After the questions, I'm going to need a 10-minute break, or my voice is going to break. Give it in before I give the lecture 9. So anybody has questions, ask questions now. Is it perfect understanding or perfect exhaustion at this stage? I'm not quite sure. Right.
I have a question.
Yes,
so
please.
I have been working with TMRM and I feel like I was doing it all wrong at this point after the lecture. But
what cells were you using?
I'm just using HeLa cells and 293 cells.
Okay.
For now. Yeah. So basically I have kind of now understood that there's like how much sensitivity you want, the speed you want. You decide which dye is good for you and which method. You would use quench or non-quench mode. But analyzing the data, a lot of people just like measure the fluorescence in the whole cell, and then they say increase is increase in potential, decrease is decrease in potential. And based on what I'm seeing, if I measure the cytosol, the mitochondria is actually the opposite. And if it's in quench mode versus non-quench mode, like how the potential is changing is totally different based on the
Yeah.
fluorescence change.
I think the value of this simulation is you can virtually design the experiment before you start. If you know what sort of thing you're looking for, are you looking for a change in mitochondrial membrane potential? For example, you can say, you can do this simulation. I mean, it won't be accurate, but you can say, should I work with rhodamine 1-3 or TMRM? Should I work in quench mode? Should I work in non-quench mode? How should I alter the loading? The relative sensitivity for the mitochondrion and the cytosol differs depending on how much probe you're loading, even when you're already in quench mode. So you can do all of those in silico experiments to optimize how you think you should plan the experiment, and then actually do it. I think that's the advantage. And you can read other people's experiments, and these dreadful ones that say the fluorescence went up by 25%, so the membrane potential went up by 25%, which is a nonsense. It's a factor of a logarithm that comes in between. So I think the value of this is use it to design an experiment in silico with your HeLa cells before you actually do the HeLa cells experiment. And you have to fiddle about a bit. As we said, always check where the quench limit is by the FCCP spike, where it just disappears. And you can save a lot of heartache, actually. Particularly,
one related point might be worth bringing up is that often when people use flow, they wash their cells before they put them into the flow system.
Yeah.
And when you're using redistributing probes, that really messes things up, particularly if you're using one with a rapid rate constant.
Yeah. So you can conceivably wash rapidly with rhodamine 1-3. In fact, you normally have to because you load with a high concentration. But with TMRM, it's catastrophic. I mean, what you would do in the simulation is you would change the loading concentration in the middle down to 0 and see how rapidly you lose probe, and it's quite shocking. Okay, any other questions here? So Ricky, I think we should have a 15-minute break now, and can I start my lecture at 30 minutes past the hour?
12:30, absolutely.
Right, okay, we'll see you at 30 minutes past the hour, as they say in America, where there are different time zones. See you later.
You guys want to go into breakout rooms to talk to each other, or do you just want to take a break and make a cup of tea?
You can put us in breakout rooms, and then people who want tea can have tea.
Can you do that, Ricky? Just put us into the same breakout rooms we were just in, and then people can leave or not as they wish.
Okay, folks, this is the last session, and I am probably feeling it rather the similar way as you are. I will share my screen. And I've done this. Really has been a sort of tradition for these courses for here and different places, that when at the end we have really a more philosophical discussion on really the state of bioenergetics and where it's going, and the state of the literature. And our general impression is that there are a lot of problems which need to be resolved. So let's actually start. This is the checklist we're going to go through in this lecture. I am focusing on nature and science and cell because they are the greatest villains in terms of scientific scientific precision and rigidity and correctness, and I will defend that statement when I give this lecture. So this is the checklist that all of us must do when we come across a paper, particularly a paper in Nature or Science, a high-profile paper written by a molecular biologist or maybe a cell biologist with an integral core, which depends on mitochondrial function. And we have to assume that a paper is guilty until proved innocent. It's the opposite of natural justice. So these are the sort of questions we ask. Do the authors actually understand the chemiosmotic theory? Are they getting confused between Gibbs free energy and standard free energy, which is just the equilibrium constant? Do they understand the laws of thermodynamics? Do they realize that things change rapidly and have to be quenched in less than a second? Do they understand with genetic engineering that if the knockout mouse comes from a different strain than the wild type mouse, you can't interpret everything? And even do they understand that causes have to occur before downstream effects? So these are mandatory, and I'm going to give you examples, each of these sort of rather egregious examples in high profile papers. Advisory, rather than mandatory, is to use physiologically relevant conditions. I mean, what I tried to do in my brown fat lecture was to emphasize as soon as possible to get as close as you can to physiology. It makes the experiments much simpler to design, much simpler to interpret. And various specificities, both Martin and I and Shona tear our hair out about ATP, people measuring ATP rather than ATP-ADP ratios at least, and coming to false conclusions. And not surprisingly, as we've just talked about, using the right conditions to measure mitochondrial membrane potential. Do the authors understand the chemiosmotic theory? So, I mean, there are examples. This is a paper, it's a letter to Nature, and out of deference to the authors, I've got rid of the title, but I've left all 30 authors there. And this was on the first page of the introduction. And interesting to sort of discuss this in the light of what we've been talking about the last 3 days. Under physiological conditions, the ATP synthase generates a proton motive force and ATP by transporting proton ions into the mitochondria. This is absolutely wonderful. If it did that, all you would need would be an ATP synthase. It would generate a proton motive force and ATP. Gosh, isn't that amazing? Let's have it's all on the same page. Under oxidative stress conditions, the ATP synthase reverses the direction, which is what I was telling you about, thus reducing ATP, cytoplasmic ATP, and the proton motive force. What have we just been talking about in terms of oligomycin null points? That the reverse is to keep the proton motive force up. Same page, this compound inhibits the reversal of the ATP synthase. It acts like the inhibitor protein, stopping it running backwards to decrease ATP hydrolysis. Yep, thereby maintaining the mitochondrial membrane potential and proton motive force. If you stop the reversal, you collapse the mitochondrial membrane potential and proton motive force. It's all in the first paragraph. There's another one. The redox potential of the mitochondrial matrix is minus 280 millivolts. What does that mean? The matrix doesn't have a redox potential. Redox couples have a redox potential. So the NADH couple has a redox potential. Disulfide thiols have redox potentials, and so on. And all of this was on the first page, and we got to the stage of half a dozen of us getting together. We wrote a rebuttal to Nature, and the editor refused to publish it, basically. Said it wasn't relevant. But we got through to them eventually, and the author said, well, you know, they're minor points. It doesn't alter our conclusions or so. And then the real shock is that this is a letter to Nature. You have 30 authors there. Not one of those 30 authors either realized the mistake or dared to talk to the PI and say, look, this is wrong. So that is egregious. The reviewers are egregious that nobody noticed that. The referees are the editors as well. And this is not a unique occurrence, and this occurs. So let's imagine a situation how this can occur. You're one of those 30 postdocs. It's a successful lab. You're publishing in Nature. You're worried that that some of the data is unsound. It's really like supporting the current US administration. You know that it's wrong, but it's in your benefit to support it. And you've just applied for an assistant professorship. And if you have this paper in Nature, your CV will be so strong you can say, I have a paper in Nature, or rather 1/30th of a paper in Nature because there are 30 authors. And what do? Do you become a whistleblower? Do you destroy the paper? You don't get the paper on your CV. Do you ruin the relationship with your PI? You probably have to leave the lab and things suffer. On the other hand, you're the PI yourself. This paper you desperately needed to get the next grant, to keep your lab funded, to keep your people employed. And you know about the problem, but without it, the whole hypothesis would collapse and you don't get your grant. What do you do? I mean, that is a real life dilemma that I know occurs frequently in these high profile papers, particularly those with multiple authors. I cannot believe that not one of those 30 authors was so ignorant about the chemiosmotic theory, they didn't realize it was totally garbage, what was written on the first page. It really is. It is shocking. I hope none of you students were among those 30 authors. I didn't actually check them, but I don't think so. I love the classical paper. I love the biochemical journal, JBC, and papers which keep to the basic idea of an abstract, introduction, methods, results, discussion, where you cannot read the results until you've gone line by line through the methods. But this is terribly unsexy. In Letters to Nature, you have the introduction of its cell together with some sexy little illustration explaining how important it is. The results, the way you get a paper in Nature, is as I'll show you in a minute, is you crowd the 4 or 5 pages with as many multi-part figures as you possibly can. The methods are totally unimportant. They're not even shown in the paper. You have to go online and find them and read them in detail to find out the methods. This is a terrible way to do science, basically. It shows that the methods are so unimportant together with, compared with the sort of the headline. And whether a paper stands or falls, it stands on the rigidity of the methods. And we've spent the last 3 days talking about numerous examples where you have to be so careful to get the methods right. The multi-part figures, this was a classic paper. It was a Sirtuin NAD paper, so it was of great interest at the Buck some years ago. And a colleague asked me, is this okay? Is this correct, this paper? And you look at the paper and it has 83 figures, 83 separate figures. And this is submitted. It has about 20 authors as well, but it's submitted, and you expect the referee and the editor to go through that paper to look up the methods for that paper, which is online, it's separate. And then you go on to this figure here, is this figure correct, and so on. And who is going to bother as a reviewer or an editor to go through all of that? And instead they simply say, oh, she's a good PI, she publishes already in Nature, it must be okay, we'll let it through. Well, unfortunately, this particular paper, a colleague asked me, can you spend a couple of days going through the paper and really focusing on the bioenergetic aspects and saying, what do you think? Is it sound or not? Do we have to believe that sirtuins affect the aging system in this particular situation? So it takes a day or two to read through the methods line by line. They're, of course, online in the supplementary data, and often they refer to a reference, and you go to that reference that refers to another reference that refers to another reference that has no validity. So it's incredibly difficult to get the methods out. But that was the summary of my report back to my colleague about some of the major errors in this paper, about some of the bioenergetic errors. So in other words, this crap gets into Nature and Science and distorts the literature and this is not me. When I gave this similar talk in Copenhagen a year or two ago, and then I managed to find a video by a clutch of Nobel laureates, including people who know nothing like Sir Paul Nurse and so on, saying that the terrible danger with nature is the publication of these sorts of papers, that people are so focused on getting this magical nature paper that they go through all these shortcuts and really dishonest steps in order to get there. And so they were very damning about the tendency to try and publish in high profile papers. I have actually 2 short letters to Nature out of my 300 and something. And the first one was written when I was a postdoc in Stockholm. And there was the World International Biochemical Congress there in 1971, I think it was, no, 72, 73. And together with a few colleagues, we looked at the entertainment, the excursions and so on. We wrote a letter saying, don't go through the official entertainment ideas. You can do the thing for half the price if you do them separately. And so that was our letter to Nature, which was complaining about the cost of the entertainments at a biochemical congress. Now, you'll not be surprised that I'm going into the curse of standard free energy. I mean, it occurs in a lot of student textbooks. Here's one example: the efficiency of cellular respiration, energy difference between glucose and carbon dioxide is 660 kilocalories, whatever that means. An ATP phosphate bond has an energy of 7.3 kilocalories. What they're saying is that the equilibrium, the Gibbs free energy, standard Gibbs free energy, which is the equilibrium constant, translates into 7.3 kilocalories. And therefore you've got 36 to 38 ATPs per glucose, 263 calories. So you're 39% efficient. Now, virtually everything there is absolutely gobbledygook. And the one thing you're going to come away from this course is never, ever using standard free energies for anything other than the logarithm of the equilibrium constant. Grievous offences against the laws of thermodynamics. As I said in my thermodynamics lecture that essentially equilibrium thermodynamics cannot prove a mechanism, but it can absolutely disprove a mechanism. If it offends against the first law of thermodynamics, you can say that it's impossible. And there was a classic example many years ago that Martin and I both contributed to. We have avoided the name so that we don't know who the head of the Burnham Institute was 20 years ago. But this was an experiment, a paper, I think it was Cell, on the early effects of inducing apoptosis in cells. And what the authors said was that an hour after inducing apoptosis, there was a 30% rise in membrane potential, and the pH gradient, the difference between the mitochondrial pH and the cytoplasmic pH, increased by about 0.8 of a unit. And if you add those sums together, see if I did it in the next slide. No, let's go previous. If you add those sums together, it means that the mitochondria have increased their proton motive force by about 100 millivolts, from 200 millivolts to 300 millivolts within an hour. Oh, I've got it up the top here. Here we are. Within an hour of inducing apoptosis. So it was thermodynamically impossible. And Martin and I, and we got a few other long in the tooth bioenergeticists to write a rebuttal to Nature. Oh, it was Nature. It's always Nature, isn't it? Which we called Apoptosis and the Laws of Thermodynamics. So we said you either believe the paper or you believe the first law of thermodynamics. They were a bit miffed about this, the authors, which included one Nobel laureate. And it was, well, forget the numbers and think about the big picture. So we had to think about the big picture there. Now, Martin touched upon this. Do the authors understand that metabolite levels are labile, that within a second or two, you need to quench a reaction when you do anything to the cell to freeze the metabolite levels so you can then analyze them at your leisure. And this was an experiment, a diabetes-related experiment with INS1 cells, where they had a mass spec, and they reckoned they could measure every single nucleotide, including AMP, ADP, and ATP. And they're doing this under two conditions: low glucose in gray and high glucose in black. And as you know, with insulin cells, they respond to glucose with a considerable increase in energy level, which is then triggering to release insulin. And the first thing you find is that it's roughly equimolar AMP, ADP, and ATP. And it doesn't change in low glucose and high glucose. So these are dead cells, basically. So the whole paper is looking at dead cells, which isn't a terribly useful thing to do it. So how did the cells die? You look at the methods. This is glucose-stimulated insulin secretion, so high glucose. The media removed, cells were scraped from the plates into a 15 mil conical tube and were centrifuged, probably for a couple of minutes. And then the pellet was frozen in a dry ice ethanol bath. That is, in the English expression, is closing the stable door after the horse has died of old age, basically. So when you spin down into a pellet, the pellet goes anoxic, and any energy-dependent changes are collapsed. What you can do, and this used to be done quite a lot, is to spin through silicone oil into, you have a multi-layer Eppendorf tube. At the bottom you have perchloric acid, if you want to to denature an acid. And above that you have silicone oil, which has a density intermediate between the perchloric acid and the incubation. You put the incubation on the top, so it's all aerobic and still functioning. You spin like the devil, and the cells wish through the thin layer of oil into the perchloric acid where they denature. And that's a possible way for doing it, not 10 minutes in a conventional centrifuge. Carrying on a similar line, some reactions are complete within a second. This was, again, an anonymous paper, an anonymized paper, dealing with another sexy subject, which is apoptosis. And this is molecular cell, okay? And they wanted to measure whether the adenonucleotide translocator was inhibited in their particular condition. So what you do, you measure the exchange kinetics of ADP across the membrane, and you can do that in the presence and the presence and absence of atractyloside, which is a potent inhibitor of the adenonucleotide translocator. And so you took paired experiments, you add the ADP to cells, to mitochondria, with and without atractyloside. It says my internet connection is unstable. I'm waiting for it to come back. Can somebody stick a thumb up if they can hear me? Can you hear me?
Yeah, you're good.
Yep, you can, good, right. And then they incubated both samples for 10 minutes before stopping the reaction, 10 minutes. And then did washing and tried to work out the counts. Now, the problem is that the ADP-ADP exchange is complete within about a second. And we know this because of experiments that the wonderful Martin Klingenberg did, gosh, is that 50 years ago? 50 years ago, he's now 90-something. And where they realized, because his great thing was the adenonucleotide translocator, and they managed to do experiments where they could reduce the time of exchange to half a second. And in ice at half a second, they just managed to detect the kinetics. These other guys used 10 minutes. So you don't get any initial kinetics when the thing has been at equilibrium for the last 9 minutes and 59 seconds. The cause happens before effect. This is a which affected me. It was a brown fat paper. and it had, again, no authors mentioned there. So I don't know who it was from the Dana-Farber Cancer Institute who did that.
You are losing your bit, David. Do you want to turn off your video to decrease your bandwidth?
Let me just stop one moment.